Welcome to our deep dive into the fascinating world of cellular scientific tools. These instruments are the backbone of modern biological research. From observing cell structures to manipulating genetic material, these tools revolutionize our understanding of biology. In this video, we will explore 12 essential tools that researchers rely on to explore the microscopic world of cells. Let's get started. Microscopes are the quintessential tools for cell observation, allowing scientists to visualize structures that are too small to be seen with the naked eye. Light microscopes use visible light to illuminate samples and can magnify objects up to 1,000 times. Electron microscopes offer much higher magnification and resolution, using beams of electrons instead of light. Transmission electron microscopes provide detailed internal views of cells, while scanning electron microscopes offer three-dimensional images of cell surfaces. Confocal microscopes use lasers and fluorescence to create sharp, high-resolution images of thick specimens. Each type of microscope has its specific uses and advantages, making them invaluable tools in the exploration of cellular biology. From basic classroom settings to advanced research labs, microscopes continue to unveil the hidden secrets of life. Flow cytometry is a powerful technique used to analyze the physical and chemical characteristics of cells in a fluid as they pass through a laser beam. Flow cytometers measure cell size, complexity, and the presence of specific molecules on the cell surface. As cells pass through the laser, they scatter light and emit fluorescence if tagged with fluorescent markers. One significant application of flow cytometry is in immunology, identifying and quantifying different types of immune cells. It is also crucial in cancer research, helping to detect and classify cancer cells. By providing rapid and precise analysis of thousands of cells per second, flow cytometers have become indispensable in both clinical diagnostics and research. Centrifuges are essential tools in the lab, used to separate cellular components based on their size and density. Microcentrifuges process small volumes, separating nucleic acids, proteins, and other components. Ultracentrifuges reach higher speeds for precise separations like isolating organelles and viruses. They generate forces up to 1 million times gravity, enabling fine separations. Crucial for biological studies, centrifuges prepare samples for various applications. PCR, or polymerase chain reaction, is a revolutionary technique to amplify specific DNA segments. PCR machines, or thermal cyclers, control temperature cycles for DNA denaturation, annealing, and extension. The process starts with denaturation, heating DNA to separate strands. During annealing, the temperature lowers to allow primers to bind to the target sequence. In the extension step, DNA polymerase synthesizes new strands by adding nucleotides. Repeating these cycles generates millions of DNA copies from a tiny sample. This precision in amplifying and analyzing DNA has transformed science. Spectrophotometers are vital instruments in the lab, used to measure the absorbance or transmittance of light by a sample. By analyzing how light interacts with a sample, spectrophotometers can provide valuable information about its concentration and composition. Different molecules absorb light at specific wavelengths, so by selecting the appropriate wavelength, scientists can quantify the presence of specific substances in the sample. In cellular biology, spectrophotometers are commonly used to measure nucleic acids and proteins. Spectrophotometers come in various types, including UV-vis spectrophotometers for ultraviolet and visible light measurements, and fluorescence spectrophotometers for detecting fluorescent molecules. Modern spectrophotometers are equipped with advanced features such as multi-wavelength analysis, high sensitivity, and automation capabilities. By providing accurate and reliable measurements, spectrophotometers are indispensable tools for studying cellular components. Cell culture is a fundamental technique in biological research, enabling scientists to grow and maintain cells outside of their natural environment. Incubators provide a controlled environment for cell growth, maintaining optimal temperature, humidity, and CO2 levels. Laminar flow hoods, also known as biosafety cabinets, create a sterile workspace for handling cell cultures. Petri dishes and culture flasks are the primary vessels for growing cells. For adherent cells, coated dishes with extracellular matrix proteins provide a surface for cells to attach and spread. Suspension cells are grown in liquid media within flasks. Maintaining healthy and uncontaminated cell cultures is critical for obtaining reliable experimental results. The tools and equipment used in cell culture play a vital role in creating the optimal conditions for cell growth. Western blotting is a widely used technique for detecting specific proteins in a sample. It involves separating proteins by gel electrophoresis, transferring them to a membrane, and detecting them with antibodies. Primary antibodies bind to the target protein. Secondary antibodies with enzymes or dyes are added. The final step produces a signal captured on film or digitally. CRISPR-Cas9 is a groundbreaking genome editing technology that has revolutionized the field of genetics. The system, originally discovered in bacteria as a defense mechanism against viruses, allows for precise and targeted modifications of DNA in living organisms. The CRISPR-Cas9 system consists of two key components, the Cas9 enzyme and a guide RNA or gRNA. The gRNA is designed to match the target DNA sequence, guiding the Cas9 enzyme to the specific location in the genome. Once there, the Cas9 enzyme creates a double-strand break in the DNA. Researchers can harness these repair processes to introduce specific changes to the DNA sequence. CRISPR-Cas9 has a wide range of applications from basic research to biomedical applications. The simplicity, efficiency, and versatility of CRISPR-Cas9 have made it a revolutionary tool in genetic research. Electrophoresis is a technique used to separate molecules based on their size and charge. It is commonly used to analyze DNA, RNA, and proteins. The process involves applying an electric field to a gel matrix, causing the molecules to migrate through the gel at different rates. Agarose gel electrophoresis is typically used for separating DNA and RNA fragments. Polyacrylamide gel electrophoresis, or PAGE, is used for separating proteins. Electrophoresis equipment includes gel tanks, power supplies, and imaging systems. Modern systems offer features like temperature control, automated sample loading, and high-throughput capabilities. By separating and analyzing nucleic acids and proteins, electrophoresis provides critical insights into molecular biology. Microplate readers are versatile instruments used to detect and quantify biological, chemical, or physical changes in samples contained within microplates. These devices are essential for high-throughput screening, enabling the analysis of multiple samples simultaneously. A microplate reader works by measuring the absorbance, fluorescence, or luminescence of each well in a microplate. Absorbance measurements are commonly used for enzyme-linked immunosorbent assays, or ELISAs, and other colorimetric assays. Fluorescence and luminescence measurements are used for detecting fluorescent and luminescent markers in various biochemical and cell-based assays. Modern microplate readers offer advanced features such as multiple detection modes, temperature control, and automation capabilities. Microplate readers are widely used in drug discovery, clinical diagnostics, and basic research. By providing high throughput and sensitive detection, microplate readers are indispensable tools in many fields of science. Automated cell counters are essential tools for accurately counting cells in a sample. They use advanced imaging technologies to provide precise counts, eliminating manual counting. The process starts with preparing a cell suspension loaded into the counter. The device captures images and uses algorithms to count cells. Some counters also provide data on cell size and viability. They come in various types, including benchtop and handheld models. Accurate counting is critical for cell culture, flow cytometry, and drug testing. Patch clamp electrophysiology is a technique used to study the electrical properties of cells, particularly ion channels in the cell membrane. Patch clamp setups consist of a sophisticated apparatus that includes a microscope, micromanipulator, and amplifier. The technique involves using a glass pipette with a fine tip to form a tight seal, or gigaseal, on a small patch of the cell membrane. By applying voltage or current through the pipette, researchers can measure the ion flow through individual ion channels. This provides detailed information about the channel's properties, such as conductance, gating behavior, and response to drugs. The insights gained from patch clamp studies are crucial for understanding the physiological and pathological roles of ion channels in cells. By providing high-resolution measurements of cellular electrical activity, patch clamp electrophysiology remains a gold standard technique in the field of cellular and molecular physiology. And there you have it. These are the essential tools that scientists use to explore the microscopic world of cells. Each instrument plays a crucial role in advancing our understanding of cellular biology. If you found this video helpful, don't forget to like, share, and subscribe for more insights into the world of science. Until next time, keep exploring and stay curious.